Good evening. I hope you are able to see the question. So, in week 11 of NPTEL course Pharmacognosy in Metabolic Engineering, today we'll discuss the sample problems. So, we start with identify the group of compounds in which resveratrol belongs. And four options are given here: stilbene, chalcone, anthocyanin, and orone. You can take a couple of minutes to think about the question. Further, we'll see how these compounds are related and what is the precursor from which these are derived. And take a couple of minutes to think about the question. Yeah, so we were looking at identify the group of compounds in which resveratrol belongs, and four options are given here: stilbene, chalcone, anthocyanin, and orone. So the first thing to do is notice structurally what's the difference between stilbene, chalcone, anthocyanin, and orone. Given that here, so this is a general structure of flavonoid where you have a benzene ring and a benzene ring connected by a three carbon skeleton and an oxygen. So if it is not cyclized, it's a chalcone. If it's cyclized, then it's a flavonone. In this cyclic group, if you have an OH, it's called a dihydroflavonol. And if this carboxy or keto group is replaced by an OH, it is a leucoanthocyanide. Now, if these two benzyl groups are connected by a two carbon uh, unsaturated group, like an ethene group, then it's called a still beam. An oron is a bit more complicated, where you have this two carbon unsaturated group. You also have this ketone group, and you also have this oxygen connected to carbon. And then we have anthocyanidin here, which has an unsaturated group here, similar to how you have flavonone, but this ketone group is not present. Now, the question was to categorize resveratrol into either stilbene or chalcone or anthocyanidin or oron. Now, all of these emerge from naringenin. And okay, chalcones are the basic structures that you get after the reaction of chalcone synthase. Now here, resveratrol's structure is shown here, where you have one of the benzyl groups, another benzyl group connected by an unsaturated two carbon skeleton. If you zoom in, this is how it looks like with the corresponding OH groups. So, yeah, we are looking at the first question. So, the question is identify the group of compounds in which resveratrol belongs. Yeah, so we are looking at the first question identify the group of compounds in which resveratrol belongs. The four options are given here stilbene, chalcone, anthocyanin, and oron. You can take a couple of minutes to think about the question and then we'll discuss structurally what is the difference and then classify it.
Is it option A, Silbin? Um, Silbin is C6, C2, C6.
Right. And the C2, is it saturated or unsaturated?
Um, that I don't know.
Okay, no issues. We had a similar question last week. Okay, we'll first look at how these are derived and how these are modified. So this is the general structure of a flavonoid, where you have a benzene group, another benzene group connected by a C3, which is uh, also having an ether linkage, meaning an oxygen combines is linking both the carbons here. Now this is derived from the chalcone structure, which cyclizes to give us the flavonoid. Now in this flavonoid, you have this double bond O. But if you have an OH in the ortho position to this double bond, it's called a dihydroflavonol. And if the double bond O, which is a ketone or an aldehyde group, if it's um, reduced to form an OH, then it's called a leucoanthocyanidin. The thing that we are interested in is the stilbene, where you have a C6 and then another C6, which is connected by a C2. And because the C2 has a double bond, which means that it's unsaturated. So that would answer that question. And then we also have anthocyanin, where you don't have this double bond O group. Instead, you have some unsaturation in the form of double bonds. And then you have an orone, which has almost everything. You have an unsaturated group here, the oxygen that links as an ether, and then a double bond O as a ketone. Right. Uh, if we look at the structure of resveratrol, it's here, where you have a C6, another C6 linked by a C2. This belongs to stilbene family of flavonoids. So it's a naturally occurring polyphenol and has been classified as phytoalexin. So that is produced in response to stress or pathogen attack. Right. So stilbene is the right answer. The next question is, how is anthocyanin different from anthocyanidin? Select the correct option. Option A, addition of phosphate moiety. Option B, addition of sugar moiety. Option C, addition of methyl group. And option D, addition of carbonyl group. You can take a couple of minutes to think about this question. We'll discuss the answer later.
Is it addition of sugar moiety?
Yes, sir. Yes, sir.
Sir, is it addition of sugar moiety?
Yeah. So what is this process called as? When a sugar moiety is being covalently attached, what is this process called as? It's called as glycosylation. Okay, you're on mute. So this process is called as glycosylation, where sugars. Okay, generally it's called as glycosylation. It depends on what specific sugar is being added. Other possible sugars could also include rhamnose. Then it will be called as rhamnosylation. So the reason for this happening is that generally these compounds do not have solubility in water, but when it gets covalently attached to a glucose moiety, its solubility increases, and it also, because of its increase in solubility, it is now able to interact with previously unknown biochemical molecules, other different proteins, and other effectors. The next question is select the correct set of anthocyanidins that consists of methyl group in their aromatic ring. Option A, pelargonidin and delphinidin. Option B, cyanidin and delphinidin. Option C, pelargonidin and malvidin. Option D, malvidin and thionidin. So you can think about this question for a couple of minutes. I have the structures of each of these in the next slide, where we'll look at which of these have a methyl group, and depending on which we will finalize the right answer. So if we look at the structures, this is pelargonidin, This
Sir,
is,
I can try.
yeah, yeah, sure.
Uh, I think that option D,
You can
that
first tell us which of the, okay, we have two molecules here with methyl group. Okay, we'll try naming each of these. That way, it will be helpful to know the subtle differences between the given molecules. Okay,
cyanidine having hydroxyl group.
almost all of these have hydroxyl groups, if you notice, because that's the
Okay.
basic structure, right? So we can't really say that only one of these has an hydroxyl group. Between malvidin and peonidin, one has one methoxy group, the other has more than that. We can think about that.
Can you show the options once?
Yeah, yeah, sure.
That option D, malvidin, peonidin, I think it is having the methyl group.
Right. It has the,
Cyanidine, dalpidin, in 3' hydroxyl, 5' hydroxyl groups are there.
okay, so this is pelargonidin, this is delphinidin, and this is cyanidin. So the difference between delphinidin and cyanidin is the lack of this OH group, whereas the difference between malvidin and peonidin is that this has one methoxy group, one OCH3 group. This has two OCH3 groups. This is malvinidin, whereas peonidin has only one OCH3 group. Okay, so this is the difference. And among the options given here, malvidin and peonidin, like you mentioned, are the ones that have a methyl group or a methoxy group. Right. The next question is, match the anthocyanidin pigments in the left side with their correct coloration given in the right hand side. In the column A, you have pelargonidin, cyanidin, petunidin, and malvidin. And in the right hand side, you have red, purple, reddish purple, and orange red. You can think about this for a couple of minutes and then we'll go to the right answer. Okay. So each of these are pigmentation molecules. And they can be categorized across the spectra. So one thing that we need to remember here is the coloration depends on the pH of the vacuole, majorly. We have a question on a related, we have a related question. We look at that as well. Okay. So shown here is a color spectra from the deepest of red to the deepest of blue. You can actually find a pattern here between so this is how it goes. The most orange is the pelargonidin, then the pinkish is the cyanidin, towards purplish is peonidin, then you have delphinidin, petonidin, and then malvidin. So the question asks for matching between pelargonidin, cyanidin, petonidin, and malvidin.
Is it option B?
1 minute. Pelargonidin, petonidin, cyanidin, and malvidin. So pelargonidin should have close to orange red, and cyanidin should have red. Petonidin should have close to blue. Malvidinin should be that case. Yeah. So if we look at this, pelargonidin has the deepest of the red, orange red. So A goes to 4, and we have option B and option D now. Others can be eliminated. Next, we have cyanidin. Cyanidin falls in the orange purple lane and not the blue one, blue spectra. So cyanidin should be close to red and not purple. So among option B and D, cyanidin is towards the purple, purple, purple red, in between both. Then we have petonidin. Petonidin is towards the blue part. C should be 2, purple. Malvanidin is the extreme blue. So it's option B. Right. The next question is identify the correct group of plant-specific alcohol acetyl transferases family in the sequence of their discovery. BAHT, ABDH, BADH, ABDH, ABHT. So this question describes how these different acetyl transferases were discovered. So this talks about a family of acetyltransferases that are specific plant enzymes. Um, so here, this has been based on the discovery. It has been named as the BAHT acetyltransferase, option A. So it's with benzyl alcohol O-acetyl transferase, anthocyanin O-hydrocinnamoyl transferase, hydroxycinnamoyl slash benzyl CoA, anthranilate, N-hydroxycinnamoyl slash benzyl transferase, and finally D-acetylvindoline 4-O-acetyl transferase. Among
Sir, what
these,
is the question?
so the question is identify the correct group of plant-specific alcohol acetyl transferases family in the sequence of their discovery. So these enzymes were discovered in plant and they belong to this class of transferases. So what is the name of this group of transferases? That's the question. So these are the different names, and here among these we have come across D-acetylvindoline 4-O acetyl transferase specifically when we were talking about Vindolin and so on. Now, briefly, if we look at them, this was the first one that was uh, uh, discovered. So this was discovered from Clarkia Brewery. Again, we did come across questions related to Clarkia Brewery as well, and it's needed for synthesis of benzyl acetate. Next, the anthocyanidin O-hydroxycinnamoyl transferase. Here, this is needed for acylated anthocyanins. Next, H stands for this HCBT enzyme, and it is required for phytoalexins. We did come across one compound of the phytoalexin, which is called as resveratrol earlier. And finally, D corresponds to D-acetyl-vindoline 4-O-acetyl transferase. This is from Catharanthus roseus and is needed for vindoline synthesis. So the right answer is option A, BAHT. Next question is, select the plant species from which D-acetyl 4-O acetyl transferase was isolated for the first time. So option A, Clarkia brewery, option B, Catharanthus roseus, option C, Gentiana triflora, and option D, Dianthus caryophyllus. We can take a couple of minutes to think about this question and then we'll answer this. Yeah, so we are talking about this enzyme, D-acetyl-vindoline 4-O-acetyl transferase, which was taken from Catharanthus roseus. It's involved in vindoline synthesis, particularly in this step. So it's called D-acetyl vindoline 4-O-acetyl transferase. So what specifically happens here is, if you see, there is an OH group here. Instead of this, you have an acetyl group. Right. The next question is, you want to create blue roses. What are the major requirements to achieve this goal?
Sir,
Yeah,
sir, 1 minute. Can you go to
yeah, yeah.
previous question? 1 minute.
Sure, sure.
Please answer.
Yeah, so we are talking about this enzyme which converts this OH from diacetyl vindoline to acetyl group. Thereby, we have vindoline. Okay, so the answer is Catharanthus roseus. That's the organism from which this enzyme was taken. Okay,
Yes, sir. Indolent. Yes.
diacetyl vindoline 4-O-acetyl transferase. It was taken from Catharanthus roseus. we'll go to the next question. You want to create blue roses. What are the major requirements to achieve this goal? One, relatively low vacuolar pH, synthesis of delphinidin, presence of flavonol co-pigments, synthesis of cyanidin, a relatively high vacuolar pH. So think about what is specifically needed for getting blue pigment and which of these among the given compounds have blue pigments, and with that we'll go to the answer. Okay, so we need to think about the colors, cyanidin and delphinidin. Okay, so let's look at this. This is cyanidin, this is delphinidin. Among these, cyanidin is towards the reddish purplish, whereas delphinidin is towards the blue colorish. So let's go with delphinidin. With that, we eliminate any options with option 4, that is option B and option D. Then we also have to talk about the pH. So now we have eliminated option B and option D. And the choice that presence of flavonol co-pigments is present in both option A and option C. So now we have to decide between the pH. Either it's low pH or high pH. So what we know is that low pH cause anthocyanidins to appear red, which means that we need a higher pH. So we need to go with delphinidin. We need the co-pigments that would stabilize and modify the colors, thereby enhancing through co-pigmentation, and we need a higher vacuolar pH. The acidic vacuoles, like I mentioned earlier, they would favor the red pigmentation over blue pigmentation. So based on this, we need to synthesize delphinidin and have flavonol co-pigments that will help in co-pigmentation, and finally have high vacuolar pH. Was it clear? The next question is, identify from the following list that is not a volatile phenolic compound? And four options are given here: resveratrol, thymol, eugenol, and carvacrol. We just looked at the structure of one of these compounds. We can just think about what would possibly be specific about the structure that would either make it volatile or non-volatile. And what is the characteristic? Okay. Okay, so here resveratrol, the compound that we looked at earlier, which belongs to stridine, it has a C6 and a C6 connected by C2. That is the non-volatile compound, and it's found in both grapes and in red. Now, because of its low vapor pressure, it is non-volatile. But whereas all the other compounds, thymol, eugenol, and carvacrol, these are found in essential oils, and they have this aromatic nature, because of which, because of their volatility, they are used in these essential oils as well. Right. The next question is, name the class of BAHT family enzymes that is responsible for the production of volatile esters in flowers. Option A, cytochrome P450. Option B, alcohol acetyl transferase. Option C, acyl activating enzyme. And option D, terpene synthase. You can think about this question for a couple of minutes and then we'll go to the right family of enzymes.
As the earlier question you told, it is alcohol acetyl transferase.
Which one? Sorry, the earlier question was about
Yes.
volatility, and we looked at
No,
the structure of resveratrol earlier, a C6 and a C6 connected by C2,
no,
and
no, no.
because
BHT families, this
BAHT,
question,
BAHT,
this
yes.
alcohol acetyl transferase, as we discussed the
Okay, you're
previously.
answering. You're directly going to the answer. Okay, okay,
Yeah.
right. Yeah, that's right. Hmm. So BAHT. includes alcohol acetyl transferases, which catalyze the formation of these volatile esters by transferring an acyl group, commonly an acetyl group, from acetyl CoA to alcohols. And they are important in the fragrance of flowers and the aroma of fruits. You're right about this. And finally, the last question is match the major floral benzenoid or phenyl propanoid volatile with their plant source. And we have three on each side: phenylethanol, benzyl benzoate, and benzyl acetate. And on the right hand side, we have jasmine, rose, and tuberose. You can think about this for a couple of minutes. Yeah, so we are discussing about these different benzenoid or phenyl propanoid volatile compounds with their plant sources. So here, phenylethanol, it is widely found in rose, Rosa hybrida. Benzyl benzoate, that is responsible for the sweet fragrance, especially in agave amica, which is tuberose. And finally, benzyl acetate, it is the component in jasmine, which gives it a sweet floral taste. I mean, sorry, a fragrance. Right. So with this, we have come to the end of today's problem solving session. We discussed the 10 questions here. I'll be on the call. If you have any other questions, we can go through that questions, or if I can clear those doubts.
So the beta oxidation pathway will be in mitochondria, na?
Which beta oxidation are we talking about? Beta oxidation of what metabolite?
The core beta oxidation pathway.
I mean, oh, what is the substrate? Starting substrate?
I think the question was like that core beta oxidation pathway. So I
Okay.
thought yeah, it
No,
is
no.
in
So,
mitochondria
so
only, na, sir?
beta oxidation, it could happen for fatty acids. I think that happens in mitochondria, so that acetyl CoA can be produced. If it's for fatty acids, then we are talking about this happening in mitochondria. Is that what you're asking about?
Yes, sir.
Okay,
Fatty acids. Yes,
fatty
sir.
acids. Yeah, yeah,
Uh,
that happens in mitochondria.
mitochondria.
Mitochondria. Yeah.
Yeah. Yeah. And the volatile you said about the volatile phenolic compound. So
Huh.
volatile fragrance of the flowers like rose will
Huh.
be due to the compounds, chavicol like compounds, or eugenol. Eugenol can be methyl eugenol, something.
Eugenol could be are found in essential oils. Not and essential oils are usually derived from the trunk and the bark rather than from the flowers, trunk, bark, and leaves.
Sir, I
Eugenol.
it got.
Yeah, yeah. Sorry, you can first repeat yourself. You were
Hello.
meant. Yeah, yeah. I can hear you. I can hear you. I can hear you, Swati. Yeah,
Hello.
yeah, yeah. I can hear you.
No, it got cut in between.
No
Yes,
issues, no
sir.
issues. So you were talking about. Are we discussing this particular question, or is it a general doubt about these volatile compounds and the sources and where they're taken from?
Yeah,
So,
the volatile you said, the thymol, eugenol, and the
huh,
clavicol
yeah,
for the fragrance of the flowers and
they
fruits, sir.
are not generally. Sorry, yeah, they are volatile. Yeah. So what's the question? I didn't get the question.
Uh, okay. So this one is carvacrol, the
Carvacrol.
volatile
Yeah.
phenolic compound.
Yeah, so here the question was which of these is not volatile and phenolic? Resveratrol, because of the low vapor pressure and molecular weight, it is not really very volatile. Whereas other three, thymol, eugenol, and carbacrol, these are relatively more volatile compared to resveratrol.
Okay.
Right.
So this will help in what, sir? Does eugenol will help in the fragrance of some flowers like that?
So all these compounds, because of their volatility, are expected to either attract insects that would help in pollination, or to be able to um, defend themselves when an insect invades it. Either of these cases. The whole point of them being volatile is they can spread easily across a larger area in a given time. So it's either for defense purpose or for attracting purpose. Like we saw earlier in the first few classes where insects should be attracted specifically to plants, flowers. So one of these purposes. Uh, was that clear?
Yes, sir.
Okay.
Sir, in the one of the this one class,
Huh.
some um, transporters were discussed for anthocyanin production, like mate, all that. So what is this mate?
N-acetyl transporters. Are we discussing about that? Or this class, I mean, week 11's lectures, is it? Which week's lectures are we discussing about?
Yeah, anthocyanin
Okay, so week
production.
11. Okay.
The yeah, some transporters were discussed.
Okay, so what exactly is the doubt there?
I wanted
I can't hear you, Swati. Yeah.
anthocyanin production
Hmm.
for the transporters which are accumulated in the vacuole.
Hmm.
Some transporters were discussed. Can you just tell about the transporters?
Okay, the specific transport. Okay. So you know that this, like we discussed about this blue roses, you need these to be produced, and this happens in specific vacuoles. For example, um, you have plastids and so on for chloroplast as well. So the first thing is these reactions happen in specific compartments in vacuoles, and in order for them to exit, you need specific transporters. So that is what you wanted to know, right?
Yes, sir. What
Yeah.
is this mate, sir? MATE?
MAT. I am not sure about it. I can look up and I'll tell you. Give me a minute.
Okay.
Anthocyanidin transporters. So based on what I've looked up, it shows that it is a multi-drug and toxic compound extrusion transporter that is generally involved in vacuolar transport of these flavonoids, anthocyanins. Again, these are
Okay.
membrane proteins that would sit on the membrane of the vacuole and allow it to get into the cytosol. So these vacuoles have something called as a tonoplast onto which these MATE transporters would sit. So these are proton-dependent transporters, and they would transport anthocyanidins into the vacuole against a proton gradient. Right.
দাঁড়া দাঁড়া বাবা জিজ্ঞেস করছে হয়ে গেছে 5 মিনিটে আসছে
Any more doubts or questions?
Yes, sir. One more
Yeah,
doubt.
sure, sure.
Um, some DFR genes,
Sorry, BFR is it?
uh,
Which this is with
DFR gene using RNA polymerase that
oh DHR is
I
it?
didn't understand. And another one, sir, what is this chalcone reductase?
Okay, we
Um,
have something called as a chalcone synthase, and we have a
uh,
chalcone isomerase. Now, chalcone reductase would convert an OH into a double bond O group in a chalcone. So this is the structure of a chalcone. Okay, this is the structure of the chalcone. And here in this pathway, we have, for example, naringenin and chalcone. If and it has multiple OH groups. So this chalcone, when being acted upon by this chalcone reductase, in this particular case, we get conversion of naringenin chalcone to isoliquiritigen. So here, what is happening is this is getting okay. So give you clarity. So this is the chalcone, and specifically in the case of naringenin chalcone, we have an extension here, which is getting reduced, and you have an isoliquiritigen here. So this is the role of CHR, chalcone reductase. Oh, is it clear, Swati?
okay, sir.
Okay. Yeah, you were mentioning about some other enzyme as well, right?
Yes, sir.
Hmm,
Another one.
hmm.
The downregulate the binary vector used to downregulate the endogenous Rose DFR gene using RNA polymerase, RNA interference, and to overexpress the DFR and F3 5 prime 3 prime 5 prime H gene for the delphinidin based anthocyanin. I didn't understand that.
Uh, do you want to share your screen? I am
Mm.
not clearly able to get the question. So you're talking about a vector, a specific vector that would help
That
in
part I
RNA
didn't get
interference,
highlight.
a specific vector that would help in RNA interference for a specific gene. So what you need to look at is like what are the options given?
Some PSPB 130, PSFL 207, PSFL 236, PSP 919, something.
Okay, so all these different vectors could be used for different purposes. In the particular case, they're talking about RNA interference. So this could be based on a particular experiment that has been described in a particular research paper. I'm not sure about the research paper, but if you can type out the question, we can check it out and go to the particular question. That will be better. Will you be able to
Okay.
type the question?
No, sir, actually I'm out of station.
Oh,
That's the
okay,
problem. I was, internet also issue is there. No problem. This one whatever you shared now, I was seeing that, sir.
okay,
Yeah.
okay. Hmm. Okay, so we discussed about DHR and this RNA interference. Right. Huh. Yeah. Other questions?
No, it's fine, sir. Yeah.
Okay. Sure, sure.
Yeah. Thanks a lot, sir. Yeah.
Thank you.
Thank you.
Yeah.